Welcome to this deep dive. Today we're actually decoding the mathematical symmetry of the human brain's origin story.
Yeah, it's a truly fascinating one to unpack.
It really is. We're pulling from this visionary, highly clinical script in the Neuromatriarch archive, and the core premise is just, well, it's incredibly humbling. Like, every thought you have right now, every reflex a doctor ever tests on you, it's all built on this microscopic architectural blueprint established by day 14 of your existence.
Right, day 14, which completely shifts how you view human consciousness, I think. We spend so
Yeah.
much time looking at the fully formed brain.
Or studying adult neurodegeneration, yeah.
Exactly. But to truly get the mechanics of the nervous system, the archive argues we have to look at its absolute origin point. Specifically, what embryologists call the week of twos.
The week of twos.
Hmm.
Because the structural symmetry of your second week in development is just, everything seems to divide into pairs, right? Your initial cluster of cells, that inner cell mass, it splits into a two-layered structure called the bilaminar embryonic disc.
Yes, two distinct layers.
You get a dorsal layer of tall columnar cells, the epiblast, and then a ventral layer of smaller cuboidal cells, the hypoblast.
And what really stands out when reviewing this isn't just the anatomy, but the destiny of those cells. That single epiblast layer, that is the sole origin point for your entire nervous system.
Every single signal and memory starts right there.
Absolutely. And at the exact same time, you have two cavities forming, the amniotic and yolk sac, to handle fluid and nutrition. Plus, the surrounding support structure, the trophoblast, also divides into two distinct layers, the cytotrophoblast and the syncytiotrophoblast.
Which, aside from being heavily tested medical terms, serve a very specific mechanical purpose for the future brain.
They do, because before the brain even exists, the embryo is already building the halt that will protect it. Those two outer layers are the vital embryonic precursors to the blood-brain barrier.
So it essentially secures the perimeter before it builds the computer.
Precisely. Without that protective interface forming first, the delicate neural tissue just wouldn't survive.
That makes perfect sense. And once that perimeter is secure, we see this massive shift in geometry during week 3. We go from a flat, two-dimensional embryonic disc to a complex, three-dimensional tube. But there's a biological leap here. How exactly do we get from that flat epiblast layer to actual neural structures?
The bridge there is a process called gastrulation. The cells of that epiblast migrate and reorganize into 3 new layers, and the outermost layer that forms is the ectoderm. Also, the precordial plate marks where the head will eventually be.
Right. And once the ectoderm is securely in place, a specialized rod of tissue called the notochord steps in. It acts almost like a biological foreman.
That's a great way to put it. It secretes specific growth factors that target the ectoderm directly above it, instructing it to thicken into the neural plate.
And this is where the physics of the development become mesmerizing.